Over the next two lectures, I'm going to introduce antibodies, their structure, their natural role in the immune system, and from my perspective, most importantly and most interestingly, how they can be exploited as drugs. I hope to convince you over these two lectures that really antibodies are one of the most interesting classes of proteins because they have so many applications, and they really are now one of the most important classes of drug molecules. So in this first lecture, I'm going to concentrate mostly on antibody structure. So aims and objectives for this first session then are to understand antibody sequence and structure, to gain an overview of the genetics of antibody diversity, and at the end of the session you should be able to discuss in detail the structure and function of antibodies and describe how the genetics allows such wide diversity. So this is just an overview of what we're going to cover in this lecture, and we're going to start with what are antibodies. They're proteins of the adaptive immune response. They act as an interface between the huge amount of variability of antigens, in other words, the molecules that are expressed on things like bacteria and viruses that invade the body. They act as an interface between those variable antigens and the constant effector molecules of the immune system. In other words, the proteins that are responsible for allowing the immune system to kill those invading bacteria or viruses. There are a number of different forms of antibody. Some of them are cell surface receptors, such as IgM and IgD antibodies. Some float around in the blood plasma, such as IgG, which is the most important class of antibody, and also a slightly different variant of IgM. Some of them are involved in inflammation, such as IgE, and others involve lining of the intestines and the mucosal tracts in order to act as a first line of attack. We'll come back to those in a few minutes. So, as I'm sure you know, the immune system is hugely complicated. I don't have time to go into any of the detail of this. I'm just going to cover the important parts of the immune system as far as antibodies are concerned. Now, this slide tries to summarize the whole of immunology as regards antibodies in a single slide. Obviously, the system is a lot more complex than this, but I'm just focusing on the key details. So, an antigen, which is a protein expressed on a virus or a bacterium, is bound to an antibody, an IgM or IgD antibody, expressed on the surface of a type of lymphocyte, type of white blood cell called a B cell. Now, this interaction occurs purely by chance. In other words, the B cell is producing an antibody which just happens to have a binding site that the antigen can interact with. The antigen then gets internalized into the B cell, where it gets chewed up into small peptides. Now, some of these peptides then get expressed on the surface of the B cell on a molecule called MHC. So MHC has a groove into which the peptide can sit. The MHC peptide complex then can interact with another surface protein, this time on a T cell, and this protein is known as the T cell receptor. When this occurs, the T cell starts to release cytokines which cause the B cell to start to expand clonally, in other words, make copies of itself. So we get more and more of these B cells that are interacting with the antigen. Now, during that expansion process, that clonal expansion, mutations get introduced into the antibody sequence. Now that means that some of these antibodies will bind to the antigen better than the original one did, and some will bind worse. Now, the ones that bind better, those will outcompete the ones that don't bind as well, and therefore undergo this process of internalizing antigen, interacting with T cell, and clonally expanding, such that we have a mini evolutionary process going on. In other words, the antibodies, or the B cells that are producing antibodies that bind effectively, will be expanded selectively compared with the ones that don't bind as well. So this process of maturation of the antibodies leads to optimization of the binding. And once the binding is sufficiently good, the B cells undergo a process known as class switching, which means that instead of producing these IgM and IgD antibodies on the cell surface, they release other classes of antibody, mostly IgG, into the blood plasma, and there those IgG antibodies can interact with the antigen, blocking its effect, which it might be involved, for example, in infection, doing a process known as opsonization, where basically the antigen is coated with antibody, triggering other cells of the immune system, such as macrophages, to ingest the cell or the antigen, and triggering other processes of the immune system, such as complement, in order to kill cells expressing this antigen. So that's their natural role. Why are they interesting to us, other than in understanding immunology in general? Well, they have a very, very high specificity for their antigen, and they also have a very high affinity, and that leads to many potential applications. They can be used as laboratory reagents, and they can be used, most importantly, as drugs. And it's estimated now that approximately 1/3 of drugs in development are antibodies, and half of the top 10 selling drugs in the world are antibodies. So they really are hugely important. So let's have a look at the composition of an antibody and its general structure before we go into some more detail later on. Before we do that, we need to clarify a little bit more about what's going on in the immune system. In the normal immune response, we produce what's known as polyclonal antibodies. So each B cell, each individual B cell, is producing one individual type of antibody. It's producing lots of them, but by clonal expansion, once this maturation phase is finished, we can produce lots and lots of identical B cells all producing the same antibody. But that process that I described could happen multiple times. In other words, we could get lots of different B cells producing each its own individual antibody, targeting the same antigen in different ways, or maybe targeting different antigens on the same virus or bacterium. Now, that's all very well, and that's what happens in the body, but if we're interested in using an antibody, we generally don't want that kind of general mixture of things. So the picture at the bottom shows you the idea of the mixture. What we want is what's shown on the right. In other words, just one individual identical antibody. And that's known as a monoclonal antibody. And these can be produced in the lab. And the way that that's done conventionally is that we harvest an individual B cell that's producing an antibody of interest, and we can immortalize that B cell by fusing it with a cancer cell line. Now, the reason for having to do that is that in the lab, B cells don't live for very long by themselves. They don't tend to divide. And so they have a quite short and finite lifetime. By immortalizing them, they live forever, and they carry on producing lots and lots of antibody. Once we've got a system that does that, of course, we can also clone the gene that encodes the antibody and then produce the antibody in cell culture. So the composition of an antibody, as we saw in a sort of schematic overview actually in those last two slides, is two so-called heavy chains and two so-called light chains. The heavy chains are approximately 440 amino acids in length and are shown in the image on the right in brown. And the light chains are about 220 amino acids in length and shown in green. So what happens is that two heavy chains come together, each of which is paired with a light chain to form a complete antibody. Now, if we try to do crystallography to look at the structure of antibodies, well, we can't look at polyclonal antibodies. They don't crystallize. Crystallization requires purity, and obviously polyclonal antibody, we have lots and lots of different antibodies, and therefore by definition it isn't pure. There's a region of the antibody which we'll come back to in a moment called the variable domain, and that's where these antibodies differ from one another when you get antibodies that bind to different things. So these are variable in a polyclonal set of antibodies. It also turns out that when we try and look at a monoclonal antibody, they generally don't crystallize either, so we can't look at those by crystallography. And the reason for this is that they're flexible. Those arms that I showed in the previous slides pointing straight upwards actually are moving around. They're flexible, and flexibility is one thing that we don't generally want when we try and crystallize things. And you can see this amount of flexibility in the electron microscope picture shown below. So you can see they all have a general kind of Y shape, but the real kind of dimensions and angles of that Y shape vary considerably between these different antibodies. However, a long time ago, a scientist called Porter discovered that the protein papain will actually cleave antibodies into two types of fragment. So this was obviously before we actually knew about the structure of the antibodies. So when he did this with polyclonal antibodies, he found that you got a 2-to-1 ratio of two types of fragment. The fragment that was present in the one part of the ratio, it turned out that that would crystallize. It was a homodimer and it would crystallize. On the other hand, the other part that was present in the 2 in the 2-to-1 ratio, that wouldn't crystallize. So what he proposed based on this information was that the crystallizable part, which he called the FC for fragment crystallizable, was going to be the same in every antibody in a polyclonal, while the other region, he thought, well, that's going to be the variable bit which binds to different antigens. So he called that the fragment antigen binding, or Fab. And he showed that that was a heterodimer. So when you do the same experiment with a monoclonal antibody, where all the antibodies are the same, it turns out, of course, that the Fab fragments will crystallize as well. Somebody working around about 10 years later, Rossi, discovered that pepsin rather than papain will actually cleave antibodies slightly differently. So in this case, it turns out that you have what's called a Fab prime fragment, which is joined by a disulfide bond, holding the two heavy chains together, or the heavy chain derived fragments. The FC fragment in this case is just slightly shorter. So it turns out that if you look at the sequence of antibody chains, and later once the structure was solved, there is some sequence similarity within the light chains and within the heavy chains. So the light chain has a kind of partial sequence repeat. The N-terminal half of the sequence looks a little bit similar to the C-terminal half. And in the heavy chains, we actually have 4 regions that look similar to one another. And so again, based on that sequence information, it was proposed that these antibody light chains consisted of 2 protein domains, while the heavy chains consisted of 4 domains. And once we look at the structure, we can see that that is indeed the case. So this is actually one of the very few antibodies that has been crystallized as a complete antibody instead of in fragments. And you can see in blue we have 4 domains working from the N-terminus at the top left of the picture down to the C-terminus at the bottom of the picture. The yellow chain is the other heavy chain, which is identical in sequence to the blue chain. So again, 4 domains working from the right-hand top of the picture down towards the bottom. And then in red, we have the 2 light chains, again, consisting of domains, in this case, 2 domains on the left and 2 domains on the right. So if we look at a cartoon view of this picture, we can see that there are a number of features that we can draw out from this. We have the 2 domains in the light chain, and these are given names, VL and CL, so variable light and constant light. In the heavy chains, we have 4 domains, and these are given names of VH, variable heavy, CH1, CH2, and CH3, standing for constant heavy 1, 2, and 3. Now, we've already come across the Fab fragment, which is shown labelled on the left here, and the FC fragment, the crystallizable fragment, which is at the bottom. Now you can see from these names that are given to these regions, these domains, that a Fab fragment consists of V domains and C domains, so variable domains and constant domains, whereas the fragment crystallizable consists only of constant domains. And that explains why the FC fragment is crystallizable, whereas the Fab fragment is not crystallizable, because in polyclonal antibodies, because it has these variable domains that vary between the different antibodies. So this region of just VH and VL is known as an FV region, so fragment variable. You can't produce this by proteolytic cleavage as you can in the other examples, but obviously with molecular biology techniques you can generate that region. At the top of this region I've shown these little red squiggly loops, and this is what forms the antigen combining site. So this is the part of the antibody that interacts with an antigen, so within the variable domains. We'll come back to this in more detail later. A couple of other features to point out. There's a region between the CH1 and CH2 which is known as a hinge. That's the region that gives flexibility, where those arms can move around. Also within each of the domains is a disulfide bond, and there are disulfide bonds within the hinge and joining the C-terminus of the light chain to the middle of the heavy chain. We'll come back to these different features in a bit more detail later. So just to reiterate the names given to the domains, the light chain has the VL and CL domains, the heavy chain has VH, CH1, CH2 and CH3. And you'll recall earlier that I mentioned there are different classes of antibodies that have slightly different functions within the body. These actually differ within their constant domains. So it's just the constant domains of, for example, an IgG is different from the constant domains of an IgM or IgE. And in fact, the IgM and IgE antibodies have an additional CH4 domain on the end here. Okay, so let's move on to the genetics of how all this happens. There are millions and millions of different possible antibodies. How can all this huge amount of variability be encoded in the genome? We know we have about 3.2 billion base pairs of DNA. Now, if we were to encode all the different antibodies in that DNA, then we'd need huge amounts more of DNA just for antibodies, and that obviously is not possible. So it turns out that there are three separate loci, three regions in the genome that encode antibody chains. There's a region called the lambda region, which encodes light chains, another region for light chains called kappa, and then a region that encodes the heavy chains. Now, if we look at what happens in the lambda light chains, it turns out that there is not a single piece of DNA that encodes a lambda light chain. Instead, we have a number of what are known as gene segments. So, picture shows there are approximately 30 so-called V segments, variable segments, each of which has a little leader segment in front of it. So don't worry about the leader. The leader actually encodes a stretch of amino acids that are used for targeting the antibody for export from the cell. Once the antibody is exported from the cell, the bit of protein encoded by that leader is removed. So the important bit to look at is just the V regions. So approximately 30 of those, and then there are approximately 4 J regions, each of which is followed by a C region. Now, what happens is that we can take one of these V regions, chosen at random from the 30, and we splice it to one of the J regions, chosen at random from the 4. Followed by its associated C region. Now this splicing occurs at the DNA level. It's not like splicing of exons that you see occurring in RNA. This is splicing in the DNA. So the DNA in the B cells is all slightly different. Each B cell has slightly different DNA from another B cell that's producing a different antibody. Now what you see at the bottom of this picture then is the result of splicing together one particular V region with one particular J region and its associated constant region. In the kappa chains, we get something similar going on, slightly different. Instead of having a number of J's associated each with a C, we have just one C region and a group of J regions. So in this case we have about 40 V's and 5 J's, and again the same sort of thing happens. We can choose one of these V's at random, one of the J's at random, and splice them together, removing the intervening pieces of DNA and following this then with the constant region, the C region. So the V and the J together in either lambda or kappa encodes that variable region that we saw at the protein level. So at the DNA level, V stands for variable and J stands for joining. And then the C region of the DNA encodes the constant domain in the protein. In the heavy chain, things are similar but slightly more complicated. So in this case, we have about 65 V regions, 27 D regions, 8 J regions, and then the constant domains. Now, as before, V is variable, J is joining, and D stands for diversity. So again, we can choose one of these 65 variable regions, one of the 27 D regions, one of the 8 J regions at random, and combine them at the DNA level. So if we do some simple maths, for the lambda light chains we have 30 times 4, that makes 120 different V domains. For kappa we have 40 times 5, that's about 200 V domains. And for heavy chains we have 65 times 27 times 8. However, we have a huge amount of additional variability because those D segments, those diversity segments, it turns out that those can go in any one of the 6 reading frames. So we have the 3 forward reading frames of DNA, and also we can read off the reverse strand of the DNA to get the 3 reverse reading frames. In addition, we have what's known as imprecise recombination. So obviously if you read in a different reading frame, you're going to have to pad the ends with extra bases. And we can get extra bases introduced at either end of the D segment, giving us a huge amount of extra random variability. So I'm estimating approximately 300 times variability there. So if we multiply together the 65, the 27, the 8 and the 300, we get about 4.2 million different heavy variable domains. And we can, in principle at least, combine those with any one of the 120 lambda chains and any one of the 200 kappa chains, giving us a grand total of about 1.3 billion different antibodies that we can produce. It's worth pointing out here that in terms of function, there appears to be no difference whatsoever between lambda light chains and kappa light chains. It's just a way of producing more variability. So overall then we have approximately 1.3 billion different variable domain combinations that we can produce by splicing together those pieces of DNA in different ways. And in fact, the antibody repertoire is far, far greater than that, probably more than 10 to the 15, because you remember from my summary of immunology slide that we get changes occurring in the antibodies. So this 1.3 billion or so that we could produce is those initial IgM antibodies and IgD antibodies that occur on the surface of those B cells. That maturation process that optimizes the affinity involves introducing additional random mutations into the sequence and therefore changing the structure of the antibodies. So I've mentioned that there are different classes of antibody, different types of antibody that have slightly different effects in the body. So let's have a look at those classes and these effector functions in just a little bit more detail. This is largely immunology, so I'm not going to go into this in very much detail. But there are 5 main classes: IgM, IgD, IgG, IgA and IgE. So IgM and IgD I've mentioned a few times. These are involved in the early response, the early immune response. So IgM and IgD are expressed on the surface of B cells. IgM is also present in a soluble form that is released into the blood plasma, and we'll come back to that in just a second. IgG is the major plasma immunoglobulin. It's a late response antibody. It starts to be produced once the affinity maturation has occurred. IgA is found in secretions, things like saliva, the lining of the gut, the mucus that lines the lungs and the trachea, and so on. And it gives a kind of first line of defence, attacking invading organisms which get into the lungs or into the throat and so on. The fifth type, IgE, is involved what's known as degranulation and histamine release, and I'll come back to that in just a moment. Now, the important thing about these different classes is that all antibodies that are produced by B cells start off as these IgMs and IgDs that are produced just by the splicing of the DNA segments. As the immune response develops and the mutations are introduced to optimize the binding of these antibodies, we get this process called class switching occurring, which allows us to produce these other types of antibody. So this occurs once the variable domains have been optimized in order to produce really good binding. We can then switch the constant domains in order to change the function. And I've listed on the right of each of these the type of constant domain that's present. So IgM has mu, IgD has delta, IgG has gamma, IgA alpha, and IgE epsilon. So how do these different antibodies carry out their functions? Well, there are specific receptors on different types of cells that interact with them. So, for example, on neutrophils, macrophages and monocytes, there are Fc gamma receptors, FcGR, which bind to IgG. And the role here is to trigger phagocytosis of pathogens that are coated or opsonized with these IgG molecules. IgE, as I mentioned, is involved in degranulation and histamine release. So there's a specific receptor, FcER, which is present mostly on eosinophils, basophils and mast cells. This triggers histamine release. So there are intracellular granules within these cells that contain histamine. When IgE is bound to these, it triggers the release of these molecules. So if, for example, you have an insect bite or a nettle sting, something like that, and you get redness and swelling occurring around the bite or the sting, that's a release of histamine from those cells causing that swelling. And the function of that is to open up those blood vessels to allow the other cells of the immune system, such as macrophages, to get to the area where the toxin has been injected through the sting or the bite. And finally, IgA has a specific receptor, the FcAR, present on neutrophils, monocytes, macrophages and eosinophils. Again, it's involved in phagocytosis, a process known as antibody-dependent cell-mediated cytotoxicity, ADCC, leading to release of superoxides and inflammatory mediators, and essentially things that will kill the target. Now, I mentioned that IgM is present as a cell surface receptor, but it's also present in the blood serum. Now, when it's released into the serum, it actually forms a multimer. It forms, in fact, a pentamer. So remember that each individual IgM molecule, like the general case we described, which was for IgG, the IgM consists of a dimer of dimers, so there are 2 heavy chains and 2 light chains, but then 5 of these come together in a pentamer mediated by a small protein rather confusingly called a J chain. Now the J chain here is nothing to do with the J segment that we saw in the genetics. The J chain, again the J stands for joining, but it's a small proteins together. And you can see in this electron micrograph on the left that there are 5 antibodies together, one at sort of 1 o'clock, one at about 3 o'clock, one at about 6 o'clock, one at about 7:30, and one at about 10 o'clock. Similarly, IgA forms a dimer, again mediated by a protein called J chain, and you can see these dimers in the electron micrograph on the left. So one of the other main effector functions, we talked about cell surface receptors such as the FCGR, which binds to IgG. Another main effector molecule is known as C1q. And C1q is the first molecule of a cascade of proteins known as complement. So C1q binding to an antibody triggers what's known as complement dependent cytotoxicity. Now what complement does, as I said, it's a cascade of proteins. So the first molecule that's involved, C1q, causes lysis of another molecule, and this cascades, activating each molecule as it goes along until it results in what's known as a membrane attack complex. So a membrane attack complex is a set of proteins which are inserted into the membrane of a bacterium in order to create a channel causing the cell to lyse. Now, to activate C1q, it needs to bind to at least 2 different Fc fragments. So in the picture on the top right here, if you imagine this is the surface of a cell and the green blobs are antigens, and we have 2 antibodies here binding to antigens. C1q, which looks like this kind of vase of tulips upside down in this picture, is interacting with those Fc regions. Now, there's been a kind of ongoing question about whether the message for how an antibody triggers C1q comes from some sort of conformational change in the antibody or from a concentration effect. Now, given that we have seen how flexible IgG antibodies are, how those arms can kind of wave around, and we saw that in an electron micrograph earlier, and from the fact that normally you can't crystallize an intact antibody, it seems unlikely that there's any way of sending some sort of conformational change through the structure. So it seems much more likely that it's a concentration effect. As we know, the C1q needs to bind to at least 2 Fcs, and if it binds to more, then it's more likely to be activated. So if you have a whole cluster of IgGs all sitting on a cell surface, this will trigger C1q and the complement cascade much more effectively. Now, because IgM is a pentamer, it's actually very, very effective at triggering C1q and the complement cascade, because you've already got 5 constant regions through which the interaction occurs with C1q. You've already got 5 of these constant regions in a group together. So overall, it does seem much more likely that it's a concentration effect that is triggering C1q and the rest of the complement cascade. So I've mentioned that what occurs in the B cells in order to generate these other types of antibody is a process known as class switching. So let's have a look in a bit more detail at how that occurs. When we look at the DNA downstream of where the V, D and J got spliced together in the heavy chain, we see a number of regions that encode the different types of constant region. So as I mentioned before, we have mu for IgM, delta for IgD and so on. Now in front of each of these, except for the delta, there is what's known as a switch site. And after each of these, other than the mu, there is a transcription stop site. So initially what happens is that we transcribe through the VDJ and through the mu and through the delta until we reach that transcription stop site. So the messenger RNA then looks like this. It encodes the VDJ, the mu and the delta. And then through conventional alternative splicing at the messenger RNA level, we can produce either IgM or IgD. Now, once we get that higher affinity, what happens is that two of these splice sites can come together in the DNA, and the piece of DNA that is intervening, in this case the bit with the mu, the delta and the gamma, gets excised from the DNA. So in this case what we have now is the VDJ followed by the epsilon and alpha regions, and transcription would occur such that we produce IgE. So the messenger RNA looks like this. It stops at the transcription stop site and we produce only IgE antibodies. Now, it's very important to realise that you cannot go back again. This is a unidirectional change. Once we've started producing, as in this example, IgE antibodies, we cannot go back to producing IgMs, IgDs or IgGs because that piece of DNA has been lost. Now, remember that we can have lots and lots of B cells which have clonally expanded and therefore are all containing the same VDJ with the same affinity, the same specificity for a particular antigen. So some of these could class switch to IgE, as in this example, so some could class switch to IgG, some could class switch to IgA. So we can have a mixture of those, and of course we can still have some that are producing IgM and IgD. We can have a mixture of all types of antibody being produced at the same time. Now, as I said, this process is unidirectional, so it can proceed forward again. So if we look at what we had before, which is production of IgE, we could bring those two remaining switch sites together, excise the DNA that encodes the IgE, and now start producing IgA. But again, we can't go backwards from that. So here's the messenger RNA that we would get in this case, coding through or reading through the V, D and J, through the alpha region that encodes the IgA, and through to the transcription stop signal. Okay, let's look in a little bit more detail now at the structure. And this is going to lead us into the second lecture. The structural detail that I'm going to concentrate on now is really just the variable domain, because that's the business end of the antibody from the perspective of having different affinities and also from the perspective of what is useful in creating drugs. So here I'm looking at the whole antibody again, the intact IgG. if we look at one of these FAB arms and zoom in on that region, here's an FAB arm. You've got the heavy chain and the light chain, and here it's interacting with an antigen. If I zoom in a bit more on now just the variable fragments, so the VH and VL domains forming the FV fragment and how they interact with the antigen. And now I take off the space filling so that you can see the backbone. So I've just joined the C alphas together. Got the antigen at the top here, and below you can see the VH and VL domains, and hopefully you can see that the region that is interacting with the antigen consists of protein loops, and these are supported on a framework which, as we'll see in a minute, consists largely of beta sheets. So this protein fold that makes up both the VH and the VL domains, and indeed all the constant domains, is known as the immunoglobulin fold. So immunoglobulin is an alternative word for antibody, but in fact this immunoglobulin fold is a very stable fold. It's known as a superfold because it's seen in many different protein families that seem to be unrelated. The immunoglobulin fold in the antibodies is a domain of approximately 110 amino acids. As I said, it's very stable, and part of that stability comes from the fact that we have these two beta sheets shown in green and red, and they're linked together by a disulfide bond, which is shown in yellow. So the views that I have on the left and right here, basically the view on the right is the view on the left, but then rotated out of the plane of the screen towards you. So you're looking from the top of the view on the left. I've already mentioned these disulfides, as well as being present within each domain, as shown by the little yellow stripes on this cartoon. There are disulfides in this hinge region linking the two heavy chains together. And there is one disulfide that links the C-terminal residue of the light chain across to the heavy chain. Now, I always say this, do not get obsessed with disulfides. Quite a common exam question will be something that involves drawing a picture like this, a representation of an antibody. And people scatter disulfides all over the place. We end up with them across joining the variable fragments together, joining the CH3 domains together. All this is completely wrong. We only have them within each domain, between the constant regions in the hinge, and joining the C terminus of the light chain to the center of the heavy chain. The other thing to notice in the domain structure, which I didn't mention before, is that there are carbohydrates. And those carbohydrates are present bound to the CH2 domains. And rather unusually, as we'll see in a bit more detail in a minute, they are on the inside of the structure. So when we look at the domains, we've concentrated on the VH and VL. Those come together to form the FV fragment, and that forms the binding site containing the CDR loops. The CH1 domain also interacts with the CL, and the CH3 domains interact with one another. In each case, approximately 900 square angstroms is buried in these interfaces. However, because of these carbohydrates that I just mentioned, the CH2 domains don't interact directly with one another. We'll come back to that in a second. Along the chain, the interactions are fairly sparse, and the linkers joining the different domains together are relatively flexible. As I just said, the interactions between the domains along the chains are fairly sparse. So what I've shown here is a fab fragment containing the VH and CH1 in red and the VL and CL in green. Now, as we rotate, at the end of each rotation, we get to the position that I'm showing in the right-hand picture. And in the right-hand picture, I've drawn a purple line through the kind of pseudo-symmetry axis between here at the top, the variable heavy and variable light, the VH and VL. I say pseudo-symmetry because obviously they're not exactly identical, but they're quite similar to one another. Then below, I've done the same between the constant light and the constant heavy one. So we could then measure an angle between those two pseudo axes, and this angle can vary. So the VHVL, the FV fragment, can flex compared with the constant regions. And this is known as the elbow angle. So, although these contacts along the chains are fairly sparse, giving us this flexibility. So I've just talked about the elbow angle, and previously I've talked about the hinge region, which allows these fab arms to flex compared with the constant domain, and that was the reason that these antibodies normally don't crystallize when they're intact, because of that flexibility. When we look between the chains, the interactions are much greater. So if we look at the variable domains, the VL and VH shown here in blue and red, they actually interact with one another via their 5-stranded beta sheets to form a barrel-like shape. Now, of course, this barrel is not actually hollow. Once you put all the atoms on and the side chains, you can see that actually it forms a really well-packed structure where the two domains interact with one another. The exception that I mentioned earlier to the good interactions between the chains is between the CH2 domains, and that's because of these sugars that interact instead and mediate the interaction between the CH2 domains. As I mentioned earlier, that's really unusual. We often get glycosylation occurring in proteins, but normally those sugars are on the outsides of proteins. In this case, they're actually mediating this interaction between the two domains. So what we have in this picture is the whole of the FC fragment, the crystallizable fragment, which then mediates in turn all those interactions with the downstream effector molecules, so binding to things like C1q and those various cell surface receptors. Just for the last few minutes then of this first lecture, we're going to look at the sequence and structure variability and how that relates to different types of antigen. So what we're referring to here is just the variable domains in the VL and the VH in the light chain and the heavy chain. Those are the bits that involve antigen binding. The constant domains, in particular CH2 and CH3, are the ones that are involved in these downstream effects. So back in 1970, two scientists, Wu and Kabat, collected as many antibody sequences as they could get their hands on. Now, sequencing at that time was largely protein sequencing. It was slow and laborious, so they didn't have a huge number of these. But what they did was to align them with one another, and they produced this plot which they called a variability plot. And what they noticed when they looked at this plot was that there were three regions just after residue 25, just after residue 50, and just before residue 100 that were regions that were more variable than the rest of the sequence. So they called these hypervariable regions, and they proposed that these hypervariable regions would actually form loops and that they would come together in the three-dimensional structure of an antibody to create what they termed complementarity determining regions, or CDRs, and that these CDRs, which is just another name for hypervariable loops, would form a binding site to which the antigen would bind. Now, this work was done several years before we knew anything about the actual structure of antibodies. This was a prediction made purely on the basis of sequence. Now, when we look at the structural information that we have, looking at the FV fragment, the VH and the VL, I've just shown you three different representations here. Top right, we have just a backbone trace, so joining the C alphas, and I've highlighted the 6 loops that form the combining site in thicker lines and colour. Bottom left, I've then taken the same sort of view and space filled those loops, and you can see that they form an almost continuous surface. There are a few little gaps where framework residues would poke through, but largely it's a continuous surface. And then on the right, I've simply done a cartoon version of that and labelled those. And the labels that I've used, L1, L2, L3, represent the positions along the sequence of the light chain. So the first loop, the second loop, the third loop, and then the same for the heavy chain, H1, H2 and H3 for the first, second and third positions on the heavy chain. Now, when you combine those two pieces of information, in other words, the structural loops that form the binding sites and that view of hypervariability that Wu and Kabat used, you see that here in the hypervariable loops, in red, green and cyan correspond to these red, green and cyan loops in the structure. The overlap between what is structurally a loop and the region that's hypervariable is not quite accurate, but certainly the hypervariable region describes what we see in terms of a surface to which antigens can bind. I've already mentioned how the V, D and J segments come together at the DNA level in the heavy chain, and the V and J segments come together at the DNA level in the light chain in order to encode the protein level variable domains. So V, D and J form the VH domain, and V and J in the light chain form the VL domain. Now what I've done here is to map where the CDR loops appear onto the DNA. So that you can see that in both the heavy chain and the light chain, CDRs 1 and 2 are encoded by the V segment. In the light chain, CDR L3 is encoded by the junction between the V segment and the J segment, while CDR H3 in the heavy chain is encoded by the very end of the V segment, the whole of the D segment, and the very beginning of the J segment. So when you look at this in comparison with Kabat and Wu's variability plot up in the top right here, you can see that the largest amount of variability was in CDR3. In fact, this is a light chain shown in the picture, but what we discover when you do the same thing with the heavy chain is that CDRH3 has a huge amount of variability, and that's because of this D segment, and as I mentioned earlier, the fact that this can go in any of the 6 reading frames, and that we get this extra junctional diversity adding additional bases in either end of the D segment. Now when we look at that in structural terms, we see that CDRH3 shown in yellow and CDR L3 shown in cyan are sitting right in the middle of the combining site. And if you want to create as much diversity in binding as possible, then you want to focus the diversity in the middle of that binding site. And that's exactly what antibodies have evolved to do. So antibodies can bind to a huge range of different molecules. They can bind to proteins, which have a relatively flat surface. They can bind to things like DNA and single-stranded DNA, which are sort of fairly linear in shape. They can bind to peptides. They can bind to sugars and haptens, which are small drug-like molecules. Now, to get that diversity of binding, you need different shapes of binding site. So we can produce a flat surface by making sure that the loops that are around the outside are relatively short and the ones on the inside, so the CDR3s, are a little bit longer. We can obtain a groove by making loops at the outside longer and ones in the middle shorter, and by making all the loops around the outside longer, we can obtain a pocket into which a small molecule like a carbohydrate or a hapten can bind. So overall, we have described antibody structure, we've described the genetics of antibody diversity, and we've looked at variability in function in terms of the different types of constant region that we can get, the different classes of antibody that produces that have different downstream effects, as well as looking at the variability in structure, which is focused in this variable domain, or these variable domains from the light chain and the heavy chain, and in particular in these CDR regions, these complementarity determining regions. In the next lecture, we're going to look at those complementarity determining regions in a little bit more detail to understand the diversity in their structures. We're going to look at the application of antibodies, why antibodies are useful, and we're going to look at the use of artificial antibody fragments and how antibodies can be used in vivo.